Thanks for coming today to listen to Ricky Wolff's talk. It's my great pleasure to introduce him. Ricky is a first-year PhD student in my lab. He just started the EEB PhD program, but he actually has spent a couple years already here at UCLA having completed his master's. And this is the work that he did during his master's. And before that, Ricky comes to us having graduated from Columbia. And it's a pleasure to have Ricky in the lab. So thank you for coming again.
Okay, so we are going to get going. Oh. Some technical difficulties here. There we go. Yeah, so on Zoom, we can see the slides, correct?
I can see everything. Okay. Don't worry about me.
Okay. Hi, so as Nandita, my advisor, who is a co-author with me on this work, along with Will Shoemaker, a postdoc in our lab, said, I'm a first-year PhD student here at UCLA. And the work that I'm going to be presenting today is on the stability of the human gut microbiome, sort of the ecological stability of the community that inhabits our gut, and sort of the appropriate level to think about that question at. So to begin with some sort of boilerplate observations that if you've gone to any microbiome talk you've heard before, the human microbiome is an ecologically complex community that has important roles in a number of health-related functions, including digestion and immune response and pathogen resistance. And of course, more functional roles are being kind of discovered all the time. But a question that is seemingly very simple, but actually turns out to be quite complicated because of the variation between different guts is what is actually a normal gut? Another way to put this is what is a healthy gut, right? So traditionally, there are sort of two ways to think about this question. And the first one I call the enterotype approach, which is a word that's been around in at least the history of in the microbiome field for a very long time, but it's a very new field. So, this is about 10, 15 years. And the idea basically is that different guts have different composition at the level of species and the level of the relative abundances of the species that inhabit guts, right? And so you can do PCA or MDS or whatever your favorite clustering algorithm is, or, dimensionality reduction algorithm is, and then cluster, say people who are obese and lean, or obese and have, irritable bowel disorder into these enterotypes, which are basically, the combinations of species that live in these people or those people, right? But another way to think about this, and one that's kind of been growing increasingly prominent as we gather more and more longitudinal data, that is data that's collected repeatedly from the same individual over, some time period, is this idea of the dynamic stability of the gut. Right, and that it may, in fact, because species oftentimes will have similar metabolic capacities, it may, in fact, actually be more important to understand how the composition of the gut changes in time and whether or not it's stable, rather than to quantify exactly what species constitute a normal gut. So you can think about, dynamic stability perhaps being a kind of criteria for what defines a normal gut, right? So we can call this the dynamic approach. So for instance, in this paper, we see that this dynamic stability is actually kind of interrupted after the administration of an oral antibiotic for Lyme disease, right? And we see that the species composition of the gut changes dramatically and then seems to actually return, relatively speaking, fairly closely to its initial baseline, right? But of course, there's variability from time point to time point just under normal conditions. The other thing, however, with the collection of more data, particularly of more sort of different kinds of data. So traditionally, much data in the microbiome field has been 16S sequencing, which is species level resolution, right? So you can tell what species are there, but species are fairly coarse, taxonomic unit, particularly in bacteria, where, of course, they trade genes with one another. So that sort of like the philosophical, epistemological use of the word is, perhaps not justified, but there's some kind of threshold cutoff. But there's no reason why this threshold cutoff actually is the ecologically relevant threshold. It just happens to be the one that the data is able to tell us about. But recently, there have been techniques that have been pioneered to get higher resolution, equivalent of like whole genome data, right? And there are a variety of techniques, but we can see whole genomes now, right? And so this has shown us that species are genetically diverse, both within and between hosts, right? So the strain of Bacteroides vulgaris that lives in me and it lives in Alex are different. And also I may have multiple strains or at least multiple, different clones of the same strain. I think as we all have if we've had any experience with, bacterial pathogens ourselves or heard about them, different strains of the same bacteria can have pretty different functions, right? Pathogenic, non-pathogenic E. coli. So it matters what strain you get. And, there are less dramatic differences in this, but, it matters what strain you get. Not all strains are alike. And there's sort of two levels at which we can think about diversity within hosts, right? There's diversity within hosts that results from independently colonizing strains reaching the host, right? And then there's diversity that is generated within hosts, right? Due, for instance, to adaptation to local conditions or neutral drift or some such process, right? So if we sort of go through and think about these processes that contribute to evolution within populations, within hosts, mutation, selection, recombination, drift. And what we know about this, we know not that much, but we know a little bit, is that there's a lot of selection, a lot of adaptation in these populations, even on relatively short timescales, right? So, you tend to see selective sweeps, which is, a new variant arises and sweeps fixation within a population on, say, 6 month to 1 year timescales in at least 10% of populations. And when you think about the population size, hundreds of billions of cells, these are impressive selection coefficients, and it's not quite sure what this is, why this adaptation is happening even within normal hosts, but it is. So if we think about kind of the number of SNVs that are affected, right? This is small numbers, 1 to 10 variants arise and they go to fixation. By contrast, we also have these processes that are happening, which we might more properly call ecological, which affect the frequencies of strains, right? Independently colonizing strains that are within the host, right? So we have 2 strains of Bacteroides vulgaris, and they fluctuate in frequency. The processes that we would, use, the words we use to talk about this are things like coexistence, competitive exclusion, neutrality. These are all words that are familiar to ecologists, but basically they just, if you're not familiar with them, they refer to different outcomes of competition between populations, right? Now, we see that there's relatively infrequently are strains showing up in hosts and then driving the resident strain to extinction, right? Particularly much more infrequently than we see selective sweeps within populations, right? Migration to the host and then displacement is much less frequent. And these processes also affect orders of magnitude more SNVs because there are lots and lots of SNVs that are segregating between strains. So for instance, if we think about 2 strains that have showed up in the same host, right? The number of SNVs that have arisen since these 2 strains diverged somewhere in the deep past, right? Tens of thousands of years ago, perhaps, is, let's say on the order of tens of thousands of SNVs. Whereas the number of SNVs that are arising within the host due to diversification, selection, that's much smaller, right? So the most important thing is that diversity is going to be dominated by the signal of these large number of SNVs that segregate between strains when there are multiple strains present, right? So we can think about belonging to a species, right? We'll have these deep fixations, right? The colors, right? Tens of thousands of SNVs, and then relatively shallow fixations or even just differences within the population. And these would be yellow stars. Good. Okay. So the last thing is that we know that hosts are oligocolonized by small numbers of strains, right? So typically the number of strains that are within a host it's less than a dozen, right? But more than just one. Now, not always more than one, sometimes it's only one. In different species, it can be different. Some species only can ever support one strain, and some species tend to always have 2 or 3. But it's always a small number of strains that are at sort of intermediate frequencies. This is at least sort of naively surprising because we have to sort of postulate a mechanism that allows 2 strains to get to the host, but not more, and then 2 strains to coexist, right, without driving each other to extinction, which may actually be sort of complicated or counterintuitive because strains should be fairly closely related, much more closely related than species, and therefore should share metabolic niches and compete with each other, right? So in order to understand why species are, or why strains are coexisting, or if they are coexisting, we need to sort of answer an even more basic question, which is how does interspecies diversity change over time within hosts, right? Are these populations genetically different time? We know that there's some evolution, but are there sort of broad patterns that that we see in terms of how interspecies diversity is changing? So the classical measure of population diversity is this thing called pi. Pi is essentially a measure of, if you were to take some random locus in the genome and then take 2 individual cells from the population, what's the probability that they would differ at that site in the genome, right? So you can see how this is a measure of homogeneity in a population, right? So if we take this quantity and we plot it, right, for a typical species within a host, and this is again in longitudinally sampled data, we chose data that was over the course of several years. So this is about 2 years, with some that are on the order of 1 year. It comes from one data set, Boye et al. 2019. But what's interesting about this data is that though it's sampled over a fairly long time scale, it's also fairly dense, right? So almost every day with some gaps. So if we look at this measure of interspecies diversity within this host, we see a sort of striking pattern, which is that though interspecies diversity tends to fluctuate, right, it tends also to be driven back to some long-term average value. This is a very striking pattern. You see it in many species, right? Now, why that would be is not totally clear a priori, though that there are explanations in population genetic theory, but it's also surprising because these values are much higher than we would expect if there was only a single strain present. So essentially, what we see here is that we have multiple strains, and these multiple strains are somehow have some constant, they're fostering some constant level of diversity within a population. So if we look at this pattern across all the species in the 4 hosts that we investigate, we see similar kinds of trends in the bulk of species. And this is a bit messy, but we can actually quantify this by doing some permutation tests. Right. Good. Right. Temporal stability over the long term. To actually measure whether these populations are not only staying at the same level of diversity, but actually changing genetically throughout the sampling period, we use a modified statistic, this pi between time points and pi between hosts, to basically measure whether the diversity that we see between time points is approaching the diversity that we see between strains that occupy different hosts. Right? So this is essentially using the between host comparison as a kind of a null for what strain dynamics could be, interpopulation dynamics could be. And what we see again is similarly, like something like 95% of species tend to stay at a, essentially the value that pi between stays at statistically the same value, right? Now there are some interesting outliers, like for instance this one, right, where we start at a value and then we see pi between hosts or pi between time points approach pi between hosts. And the reason for this, as we can see in a second, is because there's been essentially a strain replacement, right? So if we look at just if we plot all the SNP trajectories, so again, this is, we take every single nucleotide variant that shows up at any time point within this species, F. bresnitzii, and then we plot its allele frequency trajectory throughout the course of the sampling period, and then we cluster these allele frequency trajectory, these trajectories into sort of like, we cluster them and then we find basically a centroid, right? If you have enough of these, right, say on the order of thousands or tens of thousands, this is almost surely a strain, right? You see the strong clustering, all of these SNVs tend to sort of show up and to move together. Right, and the reason for this is because there's some fundamental strain frequency, right? And the fundamental strain frequency is what's determining what the value of that allele frequency is, or those many allele frequencies are, right? But this pattern is rather unusual, right? This idea that, we would start with, an initially dominant strain that would then be almost entirely displaced before making a brief comeback and then, almost surely being on its way to being replaced is unusual. Typically, what we actually see are patterns more like this, right? So this is again, we have our metric of pi, and this time it's just not plotted on a log scale, but it's the same figure I showed you before. And then we also have our metric of the strain frequencies that have been clustered, right? And so what we see if we do this clustering is that there are very likely 3 strains here, right? Now, because of the polarization, we only see the frequencies of 2 of the strains, but there is another. The third strain is there because essentially we're taking the minor allele frequency of each of these alleles, and so there is a major allele frequency that we're not seeing. We can go into that later. But sort of the more important point here is that these allele frequencies tend to remain at kind of a similar value throughout the sampling period, which is actually surprising because in population genetics, we tend to think of in terms of fitness effects, and fitness effects predict that one thing will always outcompete the other thing, right? And we see something very different from that in the real data. We see this coexistence. But quantifying this is something that we is important, but whether this coexistence is actually stable or not. And so what we did is we actually turned to a sort of a classical model from ecology, this logistic model, and we tried to fit this data to a model, right? So if you've taken even like a introductory ecology class or a differential equations class, you've seen this logistic ODE, right? And you know that there's a solution to this, but more importantly, what this equation has is 2 parameters, tau and k, right, which represent the growth rate and the carrying capacity. And the ultimate behavior of the population is entirely determined by the k, right? The population is going to be pulled to this stable attractor k. Right? So real systems are not deterministic. So we're going to fit this more complex model, the stochastic logistic model. This is a stochastic differential equation, and essentially it's a differential equation in which there's some randomness, right? Particularly represented by this term, this eta term, where we have Brownian noise, and the strength of this environmental noise is quantified by this parameter sigma, right? So basically, the width of these fluctuations, right? Good. So if we want to think about, what if we simulated this many times, we would tend to see this sort of typical behavior, right? You start at a certain value, and then you tend to oscillate around this k value, right? These fluctuations are stable, right? In the sense that they're, so in the particular sense that, in the absence of environmental noise, we pulled back to the stable attractor. In the more kind of broad sense is that there's a stationary distribution of the stochastic differential equation. And if we run this out for, for infinite time, it's gonna eventually converge to this stationary distribution, right? The stationary distribution is usually solved for. It's a gamma distribution. And so we expect if a population is really truly following this model, that it's going to have a histogram of abundances that's going to be a well described by a gamma distribution, right? So this model is actually absurdly easy to fit, right? In particular, it really only relies on 3 parameters, and we only actually use 2. We use the mean abundance and the variance in abundance, and this is the real data that. If we have these 2, relatively easy statistics, we can fit this model, and then we're going to hold this tau constant to avoid any kind of overfitting, right? So to sort of drive this point home, we don't have any free parameters here. There's no maximum likelihood estimation. We're just saying, give us the mean abundance of a population and give us the variance in abundance in a population. We can fit a model, right? So if we actually look at the behavior of real strains, right, it turns out that they're actually quite well described by this very simple model, right? So visually, we can just look at this and we say, okay, the fluctuations, which are quite large, actually, they're almost an order of magnitude. Again, these are individual strain abundances, right? Maybe fluctuating over an order of magnitude. And yet they seem to stay sort of bounded in some sense. And this combination of both the occasional large deviations and the ultimate sort of like average behavior over long term is really well described by this very simple model, right? If we then also plot the histogram of abundances, right, what we see is that this histogram of abundances also seems to converge to our expectation, right? This is this gamma stationary distribution, right? So kind of in layman's terms, what we're seeing is that these strains, when considered as sort of separate ecological units, we disaggregate them from each other, right? We treat them as what's the abundance of the strain within the community, right? We can then describe these strain abundance trajectories with a very, very simple model that is really emphasizing only that there is a sort of like constant fundamental niche size, stable carrying capacity, right? There are fluctuations, which we can determine, and populations are more or less variable, right? But essentially what we really need to do to understand like this typical behavior within hosts is this very simple model, right? Good. So if we actually look at the sort of, we develop a statistical test to test this stochastic differential equation, which essentially the way this test works is by looking at is each transition between subsequent states likely to have occurred if this model were the true model, right? So we're rejecting a null model in fact. And we see that in the vast majority of cases, despite fitting a very simple model, we cannot reject this null model. However, there are certain cases in which we can, and particularly gratifying is that the cases that we can include this species Faecalibacterium presnitzii, which I showed you earlier, which had the strain replacement, right? So when we have certain, non-stationary abundance dynamics, this test picks that up. And in the vast majority of other cases, what this test is telling us is that essentially all we need is the mean and variance in a strain's abundance to kind of fully describe what the strain is going to be doing at least over time course of years. Right. Now, if we look at all of these strain abundances, and then we consider them as kind of statistical ensembles, we can ask questions like, are there broad patterns that we can see in these broad statistical ensembles, right? And we noted two of them, right? The first is called Taylor's law, which is a well-known equation in ecology. It's one of the few sort of typical empirical laws that you see in ecology. And Taylor's law quantifies how the variance in a population's abundance scales with its mean abundance, right? Particularly, there's a power law scaling, right? This doesn't have to be, but it does tend to be in almost all ecological populations. We tend to see this, but then the exponent of this population of this law becomes important, or something that people are interested in at least. Here we see an exponent of 1.63. This is important kind of mostly in that it's less than 2, right? An exponent of 2 would essentially indicate that on a per capita basis, variance in abundance is the same for large populations and small populations, right? Now, of course, larger populations, which are larger mean abundance, will have, generically larger variance in abundance, right? But with an exponent of 2, what we tend to see is that variance is actually, it just sort of scales proportionally with the mean, right? Here, what we're seeing with this exponent that's less than 2 is that large populations actually have, they're more stable than smaller populations, right? Then we also see if we plot, again, this histogram of all strain abundances, we can rescale this histogram so that we can see it all on the same, the same axis. And then we can plot a single gamma distribution. And we see that these true histograms of strain abundance tend to collapse almost exactly to this gamma distribution, right? So, this is important partially because it's actually a prediction of the stochastic logistic model, right? Heuristically, it's important because what we see basically is that strains are tending to hang out around their mean abundance with fairly typical sizes of their fluctuations around this mean abundance, at least when we rescale everything. Right. Good. So we see these broad patterns in strain abundances that would seem to indicate that there's a kind of like a broad macroecology of the gut community at the strain level, which is perhaps unusual because we tend not to think of strains as ecological units typically in microbiome work. We tend to think of them as competing for a species level niche, right? And what we see here is that the competition for species level niche seems to occur in some species, particularly this F. presnitzii, and it's actually been found more recently that this species tends to have many more strain replacements than other species. But typically, we can actually think of strains as being at least sufficiently diverged from one another that we can treat them as sort of independent ecological units with their own stable carrying capacity that are not driving one another to extinction, whether or not they belong to the same species or different species. Right, so that's kind of the main takeaway from this work. Right. So with that, I'd like to make some acknowledgments. This is a slightly outdated picture. We have more members of the lab now, but I want to thank my advisor, Nandita Garud, who's on the call today, and also my co-author, Dr. Will Shoemaker, who's a postdoc in our lab. I want to thank as well Van Savage and Colin Kremer, who are instrumental in providing feedback for this at various points. And then lastly, I'd really like to thank members of the group lab, some of whom are here today, who have been just very helpful throughout the giving us feedback throughout the course of this paper, and telling us what works and what doesn't work. So yeah, with that, yeah, thank you.